Is here.
Yeah.
Okay, so we will conduct this course in English. Welcome to the second course of the bioinformatics. The focus of this course is to familiarize yourself on the computation of bioinformatics. And today we will cover about GATK library from Illumina. Let me share my screen. Okay. A team of Calbe AI engineer will help you with the demo of the tools called TerraBio. But let me cover the background and introduction so you can get some background before you practice some computation workflow in TerraBio. Okay, so 30 minutes for the concept and 30 minutes from practice. We want to balance between your fundamental, conceptual, and practical understanding. So gene sequencing device or next generation sequencing device is everywhere. Initially, it costs like $1 million, but now 50,000 US dollar, you can get tools like a device like Nano4 or maybe Illumina. Yeah. So Illumina is a prominent provider for sequencing platform. They sell many things like this kind of device, ISEC 100, Minisec series, next generation sequencing. Of course, there is some price if you want to get this. Yeah. And they also provide some support like tools. Yeah. You have to see the tools, trainings. Yeah. In Indonesia, in Calbe, we are partnering with the distributor of this Illumina devices, Pandu Bioscience as our partner. But we in Calbe more focus on the computational side. So you can see they also provide here some tools, but is commercial tools. What we will do in this course is much more on the open source tools. So Genomic Analysis Toolkit is an open source tools developed by Broad Institute. I think it's also funded and co-developed by Illumina because it's very specific to Illumina. So it provides you a tools to pre-process the raw sequencing data come out from the Illumina sequencing device. You can use it to align the reads of the reference genome and doing variant analysis like identifying SNPs and indels and genotyping and variant annotation and many more others use case using GATK. So before we start in GATK, we have to take a look how is the process from sample to getting the raw data from gene sequencing. So usually in the wet lab, usually pharmacy or biology, they're preparing the sample to extract the DNA or RNA from the sample like blood, tissue, or maybe saliva. Some devices use blood and saliva or other tissue from your body, and they have to prepare, they have to do extraction, okay? And library step, this is not software library, but this is the library in terms of the device, of gene sequencing device. And then they will perform reading of the sequencing reading. The machine will read the best protein like ATCG in the cluster and synthesizing the data in standard format. The output of next generation sequencing is a FASTQ file. Okay, this FASTQ file depend to the device. Some device provide FASTQ, some is already provide BAM or VCF, but most of the device support the FASTQ file. Okay, so gene database, you can find many gene database in many gene bank database like NCBI. If you read books, you can see NCBI is a major provider for the data. So many data here. So you have to learn how to get the data and how to compute using the computation platform like TerraBio that we will introduce today. So let's see on the sample preparation. Usually bioinformaticians don't do this because this is wet lab. So they have to prepare good quality sample because quality sample affect the accuracy of reading. Yeah, maybe from blood tissue and saliva. They have to prepare. They have to do some process here, like purification, blah blah blah. Yeah, so they have to also do hybridization before they put it into sequencing device. Usually, wet lab people will do this for us. We don't have to care about this until we get the FASTQ file. But during the process, they have many process here. So I just give an example here of the library of the process, like fragmentation. They fragment into smallest piece of DNA. Physical method like ultrasonification, enzymization, chemical reaction. They also do some adapter ligation and many more. Okay. I hope you read the books and make sure you know this process because otherwise some of the term you don't understand in the data. Okay. So library preparation takes fragmented or material or samples and then input and into the device, and then device will produce sequence ready file that you can do to compute. Okay. So in very high level, you have to understand this. Otherwise, some kind of a term you don't understand during your computation in bioinformatics. So there's so many reading devices. We call it high throughput sequencing now. But if you back into 1975 or 77, which is I born in 1975, 2 years from my born, Sanger method is already there. At this time, sequencing device is $1 million at cost. Even if you want to do full genomic sequencing, you have to pay $1 million this time. But becoming cheaper, cheaper, and cheaper because of the technology. So now we are more in 2024. I think it's getting more cheaper. Not only this Illumina, but also nanopore technology. There is many modern device outside Illumina, yeah? So wait, nanopore sequencing. Okay, DNA sequencing. So nanopore, 10-time genomics, Illumina is major devices. And you can see the variant of the devices is getting much, much more cheaper. Okay. So sequencing process is start by prepare the library of DNA or RNA and combine some many process like chemical, optical system to get the data. The input is actually prepared library of the sample. Okay, they call it library. Okay, because many sample they have to put. Essentially, it's a collection of fragments of DNA and RNA. Okay, and then the output usually FASTQ file. Okay, FASTQ file contain only the reads and associated quality score. So you can ask ChatGPT explain about FASTQ files and give sample of the file and explain its components. Okay, so usually it gives you the reads, the ATC, GC, blah, blah, blah. And also it gives you some score, quality score line, yeah. So you can download many FASTQ file in National Gene Bank here in National Library of Medicine, NCBI. In library, usually if you use a framework for bioinformatics, usually they teach you how to download data from NCBI Gene Bank. Data collection is very expensive. So if you are taking some topic of research in this area, I suggest you use public data bank. don't try to collect because it will translate directly to the cost. Okay, so try very much to get topic that is not require you to collect data because it take time and money. Okay, so Illumina Gene Titan platform, you can see, you can browse, you can learn many type of devices and how they read. Okay, but we are much more interested after they read it. From the output of FASTQ is the basic output of NGS, usually FASTQ file. But there's many other devices also provide aligned sequence, which is sum or bump, a sequence alignment, yeah, or binary version. We call it bump or we call it sum. Usually, some devices also generate this one. This file contain information where each reads of the genomic reads align with the reference and how well it aligns. The reference means you already know this gene, yeah, and how well it's aligned. FASTQ is not give this alignment, it's only give the quality score. But also another file like called variant identification means that you already get the variant. Okay, so you can also use ChatGPT, the same prompt to explain and give sample of this file. Let's say sum or bump, okay? And then you can also use it for VCF file. So this is two type of standard file in genomic. First is a FASTQ, is ASCII text file. Long time back, all computation happened in mainframe. Until now, there is many mainframe, S400 is still there in Japan or in any other country to perform this computation. That's why it's ASCII file like this. It's just like mainframe file, yeah. Okay, so this is sum or bump file, and then you can see VCF file. VCF already have a variant component. Please spend time understanding this component of the file is closely related with the process of sample of the wet lab that people preparing the sample. So you have to understand what is Chrome, POS, ID, blah, 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 blah. Okay. So depend to the device. If you're using Illumina or other device, sometimes they also give you gene expression and transcriptomics, yeah, also metagenomics and other specialized output. Okay, so currently gene sequencing not only for human and disease, but also there is agrigenomic from agricultural. You can find seed, you can make seed that is have better features, or you can find genomic in fish genomic, you can produce something in a food or fishery on with genomic technology. We in Calbe really believe that the future technology will be moving toward digital biology. If we say digital right now, we are much more talking about apps, database, mobile apps, web apps, IoT, AI, but the real complex digital system is biology. So that is the background, and now let's see the GATK. So it's built on Java, open source. Yeah, you can download the source code, you can compile, or you can just use it. You can use it as a library in Java, or you can use TerraBio that we will introduce today. So it provides features for data processing, like preparing raw sequence data for analysis, like FASTQ, SUMBAM, or VCF. It also provides workflow, like how to identify SNP, single nucleotide polymorphism, or indels, insertion and deletion. Also provide features for data post-processing and analysis interpretation. This is the scope what GATK provide from data pre-process, variant discovery, like workflow. There is many workflow for variant discovery and preliminary analysis for functional genomic, genotype refinement, etc. So GATK being used by Illumina technology, Illumina device. So I think they fund this project, they funded the project. So most of the Illumina use GATK and also Gene Titan technology. So there is two types of genomic sequencing. There is genome microarray like Gene Titan and also full genomic sequencing. For microarray, usually they provide some chip, yeah, electronic chip. So it identifies specific area in genomic, not full area. They only interest in specific areas, so they provide chip for the specific area. So make sure that you know what device created the file, so you can utilize library pretty well. So this is GATK features, is modular, okay, scalable. There is so many library or predefined workflow over there in TerraBio, and you can do variant discovery like SNP, indels, copy variants, and structural variants, and widely used. Okay, so let's see how GATK library. I think they have the website here. You can have the getting started here. Okay. Okay. You can do manual getting start according to this tutorial. But I think it's much better if you use TerraBio. So TerraBio have user interface, so you don't have to deal with all of this jar and do it manually in command prompt. Yeah. So you can start from TerraBio. Which is TerraBio. Okay. So TerraBio is collaboration between Microsoft, yeah, Google, Broad Institute, GATK, Illumina. They provide this. So I hope today you can learn from Calbe team about TerraBio, and so you don't have to perform manual using the library, but you have the management console web application to perform the analysis using TerraBio or using GATK. Okay. So part of the TerraBio is Dragon, Dynamic Read Analysis for Genomic. Yeah, this is the platform that's already have in some device of Illumina and supported by GATK. Yeah. So if you see Dragon, Dragon is a hardware accelerated library for pipelining and clinical application, and it's well integrated with GATK. Okay, so today I will let Calbe AI team to explain to you what is TerraBio Cloud Platform. What you have to learn in this platform is the features, the available workspace or workflow. You understand the input data, you understand the flow or workflow, what kind of analysis it can perform, and you have enough money to use it. So learn how to get the credit from Google Cloud Platform. With the credit, you can use maybe $300 to do the analysis here. And if your topic of research in this area, so you can reuse, you can continue your subscription in Google Cloud Platform for utilizing predefined workflow inside TerraBio. TerraBio also provide tutorial in YouTube. I hope you can take it as your homework for TerraBio after the presentation from Calbe team or the demo from Calbe team. Okay, so that's it, the material for today from me. I left it for the team to do some demo or discussion, or at least you can get your TerraBio account today and then say hello to TerraBio, understand how it stores data, understand the features, understand how to run pipeline, so you are more confident on your bioinformatics research. I stop here. I give it to Fadin and Sinta and other team members from Calvi to continue the course.
Okay, thank you so much, Mr. Risman, for the brief introduction of the ATK. I will continue to introduce to you for TerraBio platform. Let me share my screen first. Okay, hello everyone. My name is Fadin Safira. You can call me Fadin. I am an AI engineer specialist in bioinformatics. And today I will help you guys to get through on TerraBio and also Google Cloud Platform and how we can utilize it into our bioinformatics. So I already give you guys in the meeting chat the link that we will use for today. So this is TerraBio, as you can see. TerraBio is a bioinformatics or biological data analysis platform that developed by Broad Institute. And in Terra, you can access and analyze public data, and also we can bring your own data to TerraBio so we can analyze in here. And we can share data securely because in Terra features, they manage the secure sharing of large clinical genomic research data sets. And also we can collaborate with anyone. If you guys have some teams and have some research project, you can share your projects with your team members. And Terra is the community's choices because Terra has been used all around the world. And for across other many organizations in academic, in pharma, biotech, public health, Terra is very familiar. And if you see the features, in this tab, Terra support the analysis every month over 2 billion core hours of compute, and also for trusted sharing, and also for global collaboration. So the main aim or main goals for Terra is to provide the biomedical platform collaboration for all around the world. And you can move into this second or third link that I already gave in the chat. In app.terra.bio, and you can sign in with your existing Google account. Okay, so for the first page or the main page, we can view workspaces, view some examples in Terra, and also we can browse some data that already embedded into Terra platform. For example, let me show you guys what's in Terra. This is Terra library. In Terra library, there are data sets, feature workspace, also code and workflows. In data sets category, there are many data sets from Cancer Genome Atlas presented by National Cancer Institute or Neuroscience Multi-Omic Archive and many human cell atlas that you can guys use it for your project. And also feature workspace. On feature workspace, there are many already available workspace or workflow pipeline that already predefined by GTK that you can guys use and study what is it about. Also for code and workflows, there are some GTK for best practices workflows that you can utilize and learn. And also the workspaces. The workspaces is dedicated spaces for you and your collaborators to access and analyze data together. So there are many public workspaces that we can use and we can reproduce the workspace so we can already learn with existing predefined workspace to better understanding the concept. Okay, so Terra use a Google Cloud Platform, and you can also use the Azure for cloud computation aspect, because Terra itself is used to billings, as you can see in this section. We have to set up the billing projects for Terra, but don't worry, because we can get access to $300 from Google Cloud Platform. There is an articles that I already gave in the chat box, how to set up and how to get this $300 Google credits to explore Terra. It's pretty simple. You can scroll down and we can open link in the new tab for Google Cloud Console. And maybe I will show you guys in different account. Okay, so for your first trial, you will get the free trial within 90 days for about $3,000. You can click start free on the right top of it. Okay. So there will be the 2 steps to try the Google Cloud for free. Okay. So for this section, you have to fill in your account type, your tax information, your name and address, and also your payment method. You can use, if you have some debit cards, that Visa or credit card, it's okay to use in this, but after the trial, it will automatically no auto charge after free trial ends, so you don't have to worry about it. And I hope that you guys already prepare it, because I already mentioned yesterday in WhatsApp group. And after you click the start free, you can open your Google Cloud Console and move to the billing section. And for the billing section, you can check the checkbox. Okay, so Mr. Risman will take a break to swimming pool. Thank you so much, Mr. Risman. I will continue to introduce about TerraBio. Okay, so you have to add principal in this section. Okay. As listed in these tutorials, we have to create a Terra billing project. Add Terra strip billing at terra.bio under new principal in the form. You can copy and paste in here for the principles, and for it, select as a billing account users. So for billing account users, the roles, it can associate projects with billing accounts. So you can select save. After that, after you save, the role or principal will be appear as your billing account administrator for your email account, Gmail account especially, and also for billing account user will appear as terra-billing@firecloud.org. So it is stated in here. It is stated in here. Note that for the 2.4 step, we add terra-billing at terra.bio, but it will be appear as terra-billing@firecloud.org. So this is expected, and you guys don't have to worry about it. After that, you can create a Terra billing project into the Terra. You can click create, and there will be 2 options. You can choose Azure billing project if you guys have already subscribed to Microsoft Azure, or you can use GCP billing project that we will use for today. And you have to sign in with Google with the same account that you use as to get the $300 USD Google credit. You can type in for the Terra billing project. This must be unique and cannot be changed. So note it in here, it cannot be changed. So for example, maybe I will have write in test or maybe demo TerraBio and select billing account. So if you already succeed in creating Google Cloud billings, there will be billing account name under the name of my billing account. So we will choose that in here, select my billing account, and we will create. Okay. So the billing projects are already successfully created. Demo TerraBio billing account, my billing account. And for the new projects, billing projects will have 0 workspaces, and the member is the first account that we will use in Terra. And you can also add users to access into this billing. So if you guys have some trouble, maybe you don't have debit or credit card that suit into register in the Google Cloud Platform, you can add your friends or your team member email into your billing projects. For example, I will add myself, my other email. And you can also manage the role. Is it only users or as a owner? You can modify it later also to edit the role. And if I check into my Google Cloud, you can check it into, okay, you can move into billings. And for my projects, okay, the project will appear in here if we already clone some of the workspaces that we will work on later. Okay, I will move into the next steps after we already configure for the billing aspect. We will use the quick start workflows that provided by Terra. You can search by keyword in here. Type in quick start. And there are several type of workspaces that appear, and you can choose the Terra Workflows Quick Start. Okay, so this is the example of the workspaces that we will use today. We will learn the basics of running a workflow analysis on data in a workspace table. In this tutorial, you will practice setting up or configuring, launching, and monitoring a bug process workflow or WIDL. We call it WIDL analysis. So there will be 3 parts of this tutorial. The first part is we run a pre-configured workflow on a single sample. The second part is set up and run workflow to analyze 2 samples. And the third part is to run an additional workflow on the generated output of the step. Okay, so we have to clone these public workspaces into our spaces. To do that, you can move your cursor on the right top of the pages and you can select clone. We can modify the workspace name. For example, maybe in here I will rename it into demo Terra workflow quick start dash copy, and we have to select the billing project. I will select the demo dash TerraBio and the bucket location. So we use the default, and the description is already predefined by the workspaces author, and we can clone the workspaces. So this may take a few minutes. If you guys have some question and find some troubles, feel free to ask through chat, or you can unmute yourself and ask some questions.
Excuse me, in account critic, my NPWP number is not being identified.
Okay. So for the tax obligation main number.
This one.
Okay. So for this one, you have to type in as I will zoom in first. Hold on. Okay. For the tax obligation main number, it is character sensitive. It means that you either type in with no character like dot or strip or dash, or you can type in with dot or space or strip. Can you recheck it? Is it already correct?
Okay, I try again.
Okay. Maybe it will appear as error, but I think you have to like click your cursors into somewhere else. And yesterday I experienced it with Sinta. The first settings that the tax obligation is not identified, but after we click into some random empty area and the tax information is accepted and will not have error, I think. Maybe you can try again. Thank you for the question. Okay, let me check it first. Okay, this take a little of time. Maybe I will show you what I already created in my workspaces. So after we clone a workspaces that from a Terra global workspaces, the workspaces on the top of it will have a path to our billing. That is, I name it as test TerraBio, and also what I already renamed for my own workspaces that I cloned for the global workspaces, which is Terra Workflows Quick Start Copy. And this is indicated that I already in my own workspaces to run these workflows. Okay, if you scroll down in these workspaces, there are some quick start instruction that you can guys watch it, or you can also see the open step-by-step instruction for a better understanding. It is very complete and very intuitive also. And these third three examples is cost maybe around 15 minutes because of the processing 10 to 5 minutes per exercise, excluding time for the queue or submitted job, because it is take a more time to do the analysis in bioinformatics. Okay, we can check in our data section. There are two tables in here, sample and also sample set. Okay, this is the, I think it is error. Maybe I will test in here, clone workspace. Okay, previously I already run on these workspaces to test it, but for the raw or the initial data, it didn't have the BAM SRAM analysis ready underscore by and the other column. So it's all it's only for the my underscore sample and the NA12878 sample ID. And in analysis, okay, before that, on data, we can import data, add data to this workspace, upload TSV, open data uploader, or add reference data. So the reference data, if you click add reference data, it will appear a list of reference data that you can utilize from data on human or mouse or rat, frog, etc. And also the error data for workspaces data already embedded into this workspace. We use the ref underscore dict, ref underscore face A, and also ref underscore face A index. These three data is come from workspace data or global data. And in the file section, you can also upload your own data. And in each workspace, so for each workspaces, we'll have these 5 top menu. For the analysis, you can analyze your bioinformatics data, biomedical data into Jupyter platform, RStudio, or using Bioconductor, and also Galaxy projects. So like if you guys familiar with Google Colab, you can use in here for Jupyter to do some Python-based analysis. And for the Galaxy, if you maybe in the next chapter, we will also learn about it. And for the workflows is our goals for today, because the goal for today is to run the predefined workflows in part 1 and also run the part 2. It is similar in the part 2 and part 1, but the part 1 we only have to run it, but in the part 2 we type in a few blank column to fill in, and we can track some of the job that we submitted into the Terra in here. So onto the workflows, we can select the part 1, SRAM to BAM. So you can ask ChatGPT what is SRAM or BAM if you guys are not familiar about it. So the SRAM file is compression reference based alignment map. It's file format designed to be even more efficient than BAM. While the BAM is binary alignment map, so for these two files, we cannot view it as human read because it is require another software to interpret it, the inside of these two files. But we have to understand what this each file means. So for the BAM is binary alignment, SAMS is sequence alignment, but the BAM is the compressed version of SAM. And also the CRAM is the compression reference-based alignment map. So we have to get ourselves familiar in bioinformatics data type, data formats. And why we have to convert CRM to BAM? Because of the compatibility issue, because of some bioinformatics tools and pipeline were originally designed to work with BAM files and might not accept CRM files directly. So the CRM file it is not common to use. The more common to use is the BAM. We can do the analysis further. Yes, because in the analysis requirement, the more directly accessible in BAM format rather than CRM files, and also for the visualization and data sharing. Okay. Okay, if you guys select the part 1 CRM to BAM, you can the version will use as a default. You can also check in the source in the GitHub, and we can select a data sample from the table sample. And as you guys see in here, in each workflows, there will be the script in widget format that already predefined because we clone from the global workspaces. Here you can see the widget script that we will try to reproduce into our workflows. And from the script, as you guys can see in here, the workflow cram to BAM flow in the inputs menu, it lists as a task name and what is the variable, what is the type, like integer, string, file, etc. And also the input value. So this is already predefined. And for the first workflows, we will take it for granted to just run it. So we need the input scripts and also the outputs. Don't forget about the output. So for the output, there will be 3 additional columns in our table, such as output variable by BAM and also the report. And for that, we have to select data. Maybe we can choose this one. Okay, and we click OK. And you guys can try run analysis. Nah, and this section there will be a pop-up menu to confirm launch. Output will be saved as a workspace data in what storage, in what server in the cloud US Central 1. And you can launch analysis. This will launch one analysis. And you can track your job history in here. So when we submitted a workflow into Terra, there are some of 1, 2, 3, 4, 5, 6, 7 type of completion status that you can see. So for the queue, if we submitted a workflow, the status means that the job has been submitted to the queue, but it has not started processing yet. Maybe the system is likely busy with other tasks because we submitted a multiple of tasks and the job will have to wait its turn. And for the launching, when a job is in the launching state, it means that the system is preparing to run it. This cloud involves provisioning resources such as maybe the cloud will starting up a virtual machine, loading the necessary data into memory. And for the submitted, so you guys also notice that the status already changes from launching to submitted. Submitted, the status typically indicates that the job has been fully configured and it is ready to run. It is waiting for the necessary resources like a virtual machine to do the job. So after submitted, the virtual machine will running the job. We can track it into the job history. And once it's already done, it will change into the status into succeeded, or we can also abort and the status will change into aborted. So what is the actual output from that? You guys can see in the tables of the sample, the output is analysis ready by, analysis ready BAM, and also validation report. It will be appear in this table. So we save this output into our table in the extra columns. So for the second part, it is actually the same as the first part, but unlike the first, we have to type in the input value. Okay, maybe I will delete it first because previously I have already run it. But if you clone it, the second workflows will be in the blank columns. And you guys also can see it in the tutorial also. Okay. For the input gram, so the input value, we will utilize format like this dot something. So this means it's that the format tells the workflow to go to the root entity type of table. This means is this in this workspaces, not from the global variables in these workspaces. So the sample table, for the sample table, you guys have to look, the workflow have to look into the something that you predefined. Look into the something column to find the input for this variable. So for example, in this variable is input CRAM. So we type in this dot CRAM from the drop down menu into the input CRAM variables. The input value will be this dot CRAM. And also for the sample name, we type in this dot sample underscore ID to take the input value from the sample table from the column sample underscore ID into the variable sample name. And we have to fill in the blank ref dig, ref fasta, and ref index. If you guys remember, these three reference variable is from the global or the global data that already available in Terra. So maybe we'll type into workspaces like this, workspace dot if ref underscore this, and workspace underscore ref underscore fasta, and also workspace underscore ref underscore fasta dot index corresponding to the variable. So we have to take a look at the variables to match it into the input value. Also for the outputs, this is blank, but we can type in the output, which is this. This means that write this into our output. This dot bay, this dot BAM, and also this dot validation underscore report. And you can select data. For this, you can select the two samples. And also don't forget to save it. So the gray button will turn into blue button and we can run analysis. So the difference with the first workflow is that for the first, we only have select one sample, and for this, we will select two samples, so it will launch two analysis for one workflows. We can select launch, and it will redirect us to the job history pages, and if the status is still on the queue. Okay, we can see that the status of our workflows in here. We can monitor of all our submission in here, whether there are some tasks that is aborted, there are some tasks succeeded, or it didn't work on well, so we can monitor in job history area. And I will challenge you guys to do the part 3 to convert the BAM into UBAM, and you guys can learn it by follow the tutorial in this video, or you can guys open in here. You can see the documents of how can we run to convert from BAM to unmapped BAM. So that is pretty much it for running and configuring Terra into Google Cloud Platform, and also how to run a quick start workspaces. If you guys have some question and also want to ask something or having some troubles, you can ask in the column chat, or you can also unmute yourself and ask some question. Thank you so much. Okay, if you guys, maybe Mr. Arya have some question?
Actually, I want to ask about the um workflow. How we run it, right? Is it is it
I'm
safe?
sorry, maybe the sound it's not clear enough.
Okay.
Hello?
Is it safe for us to hello? Can you hear me? Hello.
Yeah, but
Maybe I will change it.
the sound is not clear enough.
Suaranya rada muffled, Mas.
Okay.
Maybe you can type in the chat columns. Is it safe for me to close the tab when the workflow is running? Yes, after you submitted your workflows and the job, you can track in this section whether, as you guys can see, I moved into different menu from my workspaces, or maybe different menu and also different link, if you guys can see, but the job is running in the back end. Maybe I will explain why that can happen. Okay, so this is the overview how the workflow system works. This is the back end overview if you guys interested in this area. So when Terra, we are the users of the Terra, we can work on the Terra using Terra user interface or UI. This is what you can see when access Terra in browsers. When you submitted the workflows into the Terra will take your WYSL scripts and also the inputs that we already defined into something called roles. Roles is an internal component that creates submission and sends the workflow to Cromwell. So Cromwell is a workflow management system that will take the workflows from roles and will create jobs or tasks in a simple way. And for that, the jobs will created as an individual job to send into Google Cloud Compute Engine. So the computational aspect will be uh, will be happening in the Google Cloud services, Google Cloud platform, not in our local machine. So it is safe for you guys to move into another tab, or you guys can close the browsers. It is basically what is going on in the backend. The GCP takes the job. Yeah, I think that's pretty much it. And you guys can see the submission status. It's queue launch or submitted running in here. There are several, yeah, you guys can see in here. There are several articles, documentation that provided by Terra. If you guys interested, maybe I will put it also for your own study. Yeah. Okay. Is there any other questions? Oh, okay. Yeah. I got it. So as Arya said in the chat, Arya afraid that it will terminate the process just like free Google Collect. So for my own experiment, it did not make the project into. Maybe I will close it. Or maybe I will, yeah, maybe I will close it and reopen the Terra. Okay, as you guys can see, the first part 1 demo that I submitted earlier is already done because the system already submitted and also running in the Google Cloud. But for this job is not done yet because it is still running. It takes for like 15 minutes. So don't worry if you close the tab, it will not happen like the process in Google Colab, I think. Oh, so there is a question from Sinta. Do we need to turn off the GCP? Okay, for the GCP, as you guys can see in here, I think. Okay, total cost. You can see in here, total workflow runtime charges, Google Cloud Services cost for all these 3 exercise is cheap, should be much less than $1 USD. Cost for running the workflows and storing output in the workspace bucket will be charged to the TerraBio project. You assign when you clone your own copy of the workspace. I think it is, you will get charged for running in for the workflow runtime, and I think it will they will cost us as the runtime is in the running, but when it's already done, the cost it will not charge to us. you can check it in here, especially when you want to run maybe in here. Okay, we can modify in the workflows. You can try and discover it maybe for the Jupyter create analysis. We also can create like Jupyter Cloud environment. We can customize it, like what type of CPUs that we want, like two memory for core CPU, one core CPU, etc. And of course the cloud compute cost will be different for running per hour. So make sure when you're running like Jupyter Cloud environment in this in the TerraBio, you will notice how much hour that you will actually run it. But for today, our goals is to run these two parts, and you guys can, I challenge you guys to do the third part to run an additional workflow on the generated output of the stack. So you guys have to run it the second part first, and the input for the output from the second part will be the input for the third part. You guys can experience it. Okay, thank you so much, guys, for your participation in these classes. We can meet up again next week for another topics. And bye-bye. Have a nice weekend. Thank you so much.
Thank you, Mbak Fadin. Thank you.
Thank you.
Thank you. Terima kasih.
Thank you.